Hi, this is Science Hangout from Science Mission. So I'm Sadashiva Pai. So we have today Dr. Raul Mostoslavsky with us. So he is an associate professor at Harvard Medical School, and he has several articles, very seminal publications in different journals. So he has won several awards. In 2017, he won a Premio Recis Award for International Cooperation from Ministry of Science, Argentina. He was awarded Laurel Schwartz Endowed Chair in Oncology from Harvard Medical School, plus Foundation Prize on Excellency in Hematology Oncology Research. In 2012, he won MGH Research Scholar Award. 2011 Honorary Guest Award from Government of Tucumán, Argentina. And he won several awards in 2009, 8. The list goes on and on and on. So I think I have to stay here forever to say all these awards. Anyways, so let me introduce Dr. Mostoslavsky. Dr. Mostoslavsky, welcome to Science Hangout.
Thank you, Sadashiva. It's a pleasure to be today and be able to tell you a little of the science we are doing in my laboratory.
So just to start with, just tell us about yourself rather than me introducing you more. It's better to hear from yourself. So go ahead and talk about yourself.
So I study medicine in Argentina. I'm from Argentina, from a small city in the north of Argentina. I studied medical school. When I was finishing medical school, I decided I wanted to do research. I was very curious about understanding mechanisms, and I didn't feel that I was getting a more understanding of the mechanism by doing medical school, so I decided to go for a PhD. I went to Israel. I did a PhD in molecular biology in the Hebrew University of Jerusalem, and then moved to the States. I moved to the States for my postdoctoral fellowships. I was at the Children's Hospital in Boston, here in Boston, where I still was working on immunology and chromatin accessibility. And while working on chromatin, we came upon this novel group of proteins called SIRT6. And when I was finishing my postdoc, I was already heavily involved in trying to understand the functions of these proteins, SIRT6. And that's what my lab has been doing since I started in MGH, in Mass General Hospital at Harvard. It's been 10 years since I've been in my own laboratory. And we've been using one of the SIRT6 called SIRT6 as an interesting starting point to understand different biological processes, as I will tell in a minute in my presentation.
Okay, so in your research career, do you have any mentors, role models?
I did, actually. I was very fortunate and lucky to have excellent mentors throughout my career. Starting on my PhD, I did my PhD commented by two people, Judith Bergman, she's an immunologist, and Howard Cedar. He was a pioneer in chromatin biology. Both excellent scientists and fantastic mentors. They really pushed me to try to understand better, to make the right questions. They really motivated me to go to the lab every day. And then I moved to the States, where I was also fortunate to be as a postdoc in the laboratory of Fred Alt. He's a Howard Hughes investigator in the Boston Children's Hospital, an amazing scientist working on basic fundamental mechanisms of immune receptor. And again, Fred was also an amazing mentor, so passionate about science. Every day coming to the lab was really a pleasure. We came to try to understand mechanisms, and I think from the three of them, I got a fantastic mentor guidance in how to be myself a PI in a lab.
Okay, and I know you are busy with all your work, research and all. So do you do anything else? Can you tell us about your hobbies?
I do. I think that as scientists, we spend so many hours in the lab every day that if you don't try to balance when you are outside the lab, if you don't try to balance, then you will be miserable. And I think it's toxic. You do need to to try to balance. And I do. I have a very active life outside the lab. We love travels. With my wife and daughter, we travel a lot when we have time. And I like scuba diving. So when we travel, I try to go to places where I can scuba dive. My wife is a marathoner. She runs. So we also find places to travel where she can run a marathon and we can go with her and trek with my wife and find the new places. I also play tennis once a week with my brother. So I try to keep it busy also outside the lab.
Okay. Now coming back to the lab, tell us about your lab, your students, a little bit briefly about everything.
So I have now a medium-sized lab with 8 people. I have a lab manager, 4 postdoctoral fellows, and 3 graduate students. So it's very balanced. I have both students and postdocs. I've been very lucky since the start of my lab. I got excellent people to join the lab, all very motivated, clever, self-motivated. They always have people that challenge me, that will come with questions, that they will read and come and we'll discuss. So it's been a very fruitful lab environment since the beginning of my laboratory. You ask me how I keep them motivated. I think they basically, I don't think you can really enforce that in a person. Most of the people that apply to my lab, they come very self-motivated and they see me. They see how much I enjoy coming to the lab, how much how much effort I put in the lab, how much passion I put in the lab. And I think this reflects out when a student or a postdoc see your PI that it's fully engaged on the work you are doing. They also are motivated. I think this comes naturally. I don't think you can enforce these things. I always tell someone that is joining my lab, I hope you come really motivated and you think that what you are doing is amazing work. If you don't feel like that, this is not the right career for you, because science is a career where we exposed to many bumps in the road. For every experiment that works, 10 other experiments don't. So you have to be a pathological optimist, as I call it, because you really need to grasp those few experiments that work and trying to understand the science behind it, because for many days things won't work, so you need to keep trying and persevere. So that's what I tell when people join my lab. You have to be passionate, you have to be persevere and really have a positive attitude, otherwise it will be a very difficult career choice.
Okay, and you developed these mouse models. I think I'm getting some info. Mouse models in your career. Like, how do you went on with developing,
So
getting other things?
this was at a time where we knew very little about these SIRT6. This was a group of a family of proteins that were identified in yeast and with potential of work as chromatin modulator, using as a cofactor a molecule called NAD, which is a metabolite. So people immediately realized, maybe because it's using a metabolite as a cofactor, these chromatin factors could be influencing metabolic decisions at the level of the chromatin. We knew very little about them, and I was in a laboratory where we make mouse models to understand function. So rapidly, at the beginning of my postdoc, we said, okay, we want to understand how these SIRT6 function. Let's make mouse models for them. And there were 7 homologs in mammals. We knew very little which one to follow at that time. So I ended up making mouse knockouts for many of them. And it was at the right time, because it was a field that was exploding. Many people were interested on those proteins, and we were the first to make the mouse knockouts. So very early during my postdoc, I was already sharing these reagents with many laboratories. And I think it was the right time to be doing these experiments, because it's a lot of people were interested on these proteins. By the time I moved to my own lab, I was already collaborating with many people around the world for these animals.
So let's hear about your work now. Please go ahead and share your slides. Thank you.
Let me see. It doesn't let me share only the application. It says share the screen. Hold on a second. Tell me if this is okay. No, this won't work.
No.
How I stop sharing?
Unshare again, double click on. Yeah.
Okay, it doesn't let me share the application only. I don't know for what reason.
Do you get different windows?
Yeah, I do have 2 different windows. Hold on a second. I will try to do this by sharing a window instead. One second. Let's see if this works. Tell me if this works. Is that okay?
Yeah. Yeah.
This looks all right?
Yeah. Go ahead.
Okay. Okay, so we'll try to do it like this. So as I said, my laboratory worked with this protein called SIRT6. It's a unique factor among the 7 mammalian homologs. What SIRT6 does is it's what we call an histone deacetylase. It's an enzyme capable of removing acetyl groups from proteins. And you can see here in the presentation, it's just a diagram. That's when SIRT6 is present, it removes these acetyl groups from chromatin. And acetylation in chromatin usually it's linked to active marks. And when SIRT6 remove those marks, you basically compact chromatin and you express gene expression. And on the right, you can see a Western blot where if you remove SIRT6 by knocking down SIRT6 in hours, you see this massive increase in K56 and K9, which are the 2 lysins that SIRT6 regulate. So what we identify SIRT6 as an histone deacetylase. This was originally done by different groups that biochemically show that SIRT6 can remove these marks. We were interested in understanding the biology. What does SIRT6 regulate in tissues, in cells, and can we understand better the biology by modulating SIRT6 levels? So what we did, and this is just from a review we wrote a few years ago. Now that's 10 years of research in SIRT6. It plays many roles in multiple systems. Here I just show a summary of SIRT6 as an histone deacetylase, control expression of glycolytic genes affecting glucose metabolism. It controls expression of inflammatory signaling, influencing inflammation, lipid metabolic genes. It influences cancer by many ways, and I will show you examples from our own work, etc. It's really a multifunctional, polyphacetic protein with multiple biological roles. So we made the mouse knockouts many years ago, and what the animals develop, among other things, the main things these animals develop is this profound hypoglycemia. So consistent with these proteins using NAD as a cofactor, not surprisingly maybe, SIRT6 knockout animals had these major metabolic abnormalities. What was for us a surprise is that SIRT6 was unique among the different sirtuins, developing this very acute degeneration. The animals were born normally at Mendelian ratios, but in days, you see that the animals started losing weight, and this on the right here below, this is glucose levels in blood. You can see that in days, the glucose goes down, plummet down, to a point where by day 23, 24, every animal was dying from a coma hypoglycemia. Wow. So it was a major severe phenotype, this severe hypoglycemia, and we really didn't understand at the time why lack of this unique chromatin factor gave us this metabolic phenotype. So when I was starting my own lab, I moved here to Mass General, we decided to tackle this question and try to understand why lack of SIRT6 caused this change in glucose. And what we found, just to make a long story short, we published this a few years ago, what we found was that SIRT6 is a repressor of glycolytic genes. It sits on multiple metabolic genes, particularly glycolytic genes, and it represses a factor called HIF-1 alpha. And what we found is that actually in most of the cells in your body, which are not rapidly proliferating, the presence of SIRT6 put a check, a brake on these glycolytic genes. So most of the pyruvate that goes into the cells goes to the mitochondria to produce ATP through the Krebs cycle, through the TCA cycle. And that's what the most efficient way to produce ATP, and that's what most of the cells in your body will do. So SIRT6 is in charge to putting a brake on these genes so you keep normal mitochondrial respiration. Now, conditions of nutrient stress, low glucose or hypoxia, low oxygen, where the TCA cycle cannot function properly, made those cells go through what we call a glycolytic switch. SIRT6 was released from the, and I can show you here, I don't have the animation, but basically SIRT6 is removed from proteins and you get acetylation in these genes, basically causing a switch. You start expressing these glycolytic genes, and among them we have lactate dehydrogenase, pyruvate dehydrogenase kinase, the glucose transporter, and you make a switch where the cells start uptaking much more glucose but producing lactate, less efficient to produce ATP, but gives the cells an opportunity to survive. And the reason why the animals were dying was because few tissues, skeletal muscle and brown fat particularly, were actually uptaking glucose in an uncontrolled manner. So we removed this chromatin factor, and constitutively, these animals have uncontrolled uptake of glucose. The glucose was going down in the blood, and basically the animals were dying because the brain didn't have enough glucose to survive. So we proved this in normal cells. That was the function of SIRT6 in normal cells as a critical regulator of this glycolytic switch. Depending on how much SIRT6 was sitting on chromatin, the cells will decide how much of the pyruvate will go to the mitochondria or will stay in the cytoplasm to produce lactate. When we were finishing this experiment, we realized that when we removed SIRT6 from the system, we were making these cells constitutively glycolytic. They were producing lactate in an uncontrolled manner. And this was independent of the nutrient or oxygen conditions. This SIRT6 molecule was so dominant in the phenotype that the moment that when we remove SIRT6, they didn't care about the nutrient conditions. They start producing lactate because they didn't have this repressor in the system. So we wonder, this phenotype of lactate metabolism, even in the presence of oxygen, it's a phenotype that was described many years ago in cancer cells. And this was described by an amazing scientist called Otto Warburg. We call this now the Warburg effect, where cancer cells basically shift towards aerobic glycolysis and production of lactate, even in the presence of oxygen. That's why we call it aerobic glycolysis. And this was precisely the phenotype of SIRT6 deficient cells. So we hypothesized maybe tumors get rid of SIRT6 to acquire an increased metabolic advantage. In other words, maybe SIRT6 is acting as a tumor suppressor. Maybe it's acting as a tumor suppressor to protect cells from this glycolytic switch. So we started exploring this in different models, and a few years ago, we published a paper showing that at least in the context of colon cancer, SIRT6 acts as a very robust tumor suppressor. So we made animals that were conditionally deleted for SIRT6. So we removed SIRT6 specifically in the intestine. Remember, the full knockout died too early from the hypoglycemia. So we couldn't use the full knockout. We developed an animal where we deleted SIRT6 only in the intestine, and we crossed to a model called the APC-mean mouse model of colon adenomas. APC-mean alone in the mouse give you only adenomas, never invasive. When we crossed to the SIRT6 animal, we got much more tumors, bigger tumors, and actually adenocarcinoma. You can see here, this is an invasive adenocarcinoma. So by removing this chromatin factor, we made the tumors much more aggressive. And what was more interesting, and I will move this here, the tumors, when we treated with the drug called dichloroacetate, DCA, the tumors were, this is a drug that inhibits glycolysis. When we give them to the animals in drinking waters, look here on the right, these two are the panels of the SIRT6 knockout animals. Most of the low grade was gone, all of the high grade were completely disappeared. So it was, this told us two things. SIRT6 as a chromatin factor plays a key role in protecting against this metabolic switch. And when you don't have SIRT6, tumors that manage to eliminate SIRT6 become addicted to this glycolytic switch. If you inhibit glycolysis, the tumor cannot grow. Okay? So that's basically what we proved in that paper. It was the first demonstration of how a chromatin factor could modulate this metabolic feature of tumors. So this was published some years ago. We then decided to ask, so that's basically a summary of that paper. We show that SIRT6 as a chromatin factor, when tumors eliminate SIRT6, you don't need to acquire what is called second hits. And these tumors actually that were deleted for SIRT6 show no activation of oncogenic signaling, RAS, ERK, because it was enough to get rid of this chromatin factor to acquire this very aggressive transformation. So at that time, we asked two main questions that we follow. The first was, how early is this glycolytic switch established? Can you listen? Okay.
Yeah, yeah.
So the first question was, how early is this glycolytic switch established? Is this a late adaptation of these tumors, or is it something that is acquired early during the process of transformation? And the second question was, are there other cancers where SIRT6 function as a tumor suppressor? So for the first question, most of the people in the field believe that this is a late adaptation, that tumors that grow very big, they start acquiring areas of hypoxia, necrosis, so you need wallboard to maintain biomass and sustain this rapidly proliferating of highly aggressive tumor. So a lot of people in the field really believe this was a late adaptation. We have some data that suggested that this is not a late adaptation, but rather a very early one. And I will just show you one of these data. If you look here on the right, this is colorectal cancer data set from the NCI, showing very low levels of SIRT6 in the cancer compared to normal tissues, and this is concomitant with the upregulation of the glycolytic genes. Now, in colon, you can look at stages. And if you look at the stages in the colon, even at stage 1 colon carcinoma, already the cells were downregulating SIRT6. So this told us that this is not a late adaptation, and suggested to us that maybe tumors very early during the process of transformation were acquiring this metabolic feature of the Warburg effect. So we started looking at this, and this is unpublished data, which we are still working on it. What we basically see is that in the intestinal colon carcinoma, you have very good models to look. There's an investigator called Hans Klevers who showed that the precursors of the cancers in intestine are these cells that sit in the crypt of the intestine called intestinal stem cells. These intestinal stem cells that are marked by a marker called LGR5. They sit on the base of the crypt, and what Hans Klever shows is that if you mutate APC, this is the same gene I mentioned before that can drive intestinal tumorigenesis, exclusively in these stem cells, you get full-blown adenocarcinoma. If you do that in the transitional amplifying cells, you get nothing. So really, these intestinal cells, intestinal stem cells, are the one that will be, if they mutate APC, will become intestinal cancer stem cells and will drive the phenotype. So we ask, maybe those are cells that are glycolytic, and if you don't have SIRT6, you increase glycolysis, you increase the number of these cells, and that's why the animals end up with more tumors. And we had a perfect system to look because we have made those animals that were SIRT6 APC mean. So we started asking those questions, and we're still working on this, but basically what we see is first, in the absence of SIRT6, if you count those intestinal stem cells, we see an increase in the number of the stem cells. And you can see here is a 30% increase, a clear increase in the number of the stem cells, and we rescue that by inhibiting glycolysis. So this tells us that in the absence of SIRT6, we do increase the number of the cells, and this is dependent on glycolysis, because when we inhibit glycolysis with this small drug, the increase in cells goes down. Now, we could also look, there is a way to grow these cells in culture in what is called intestinal organoids. So you can grow 3D models of the intestine, and the number of organoids you get is a direct reflect of the number of stem cells that you started the culture. And here you can see on the left, in normal intestinal organoids, you see the same increase we saw in the intestine, suggesting that indeed we have more stem cells. But if we do this from an animal that is already transformed with APC, then the increase is much more severe. We have a threefold increase in the number of organoids, suggesting that in the context of a transformation event, the increased glycolysis plays a much more critical role. And we also were able to rescue it by inhibiting glycolysis with the dichloroacetate drug that I am not showing it here. So we try to prove that indeed these stem cells are more glycolytic. We try first using an animal that is a GFP reporter from the LGR5 cells. This is not a perfect model because the GFP positive cells are chimera, and the Cre recombinase doesn't fully activate in the GFP cells. So you can see some increase in glycolytic genes here, but not in all of the cells. So we decided to find a different model. We created one. What we did is we generated a reporter where we can, in the organoids, express by lentiviral expression, a gene where we had the promoter of this glycolytic gene called PDK1. It's one of the genes that were highly upregulated in the knockout, with a reporter in cherry, and a second reporter with a marker of the stem cells called OLFM4. And when we did the double staining, we got a very interesting surprise. So this glycolytic expression, you can see it here in red. It's not expressed with difference quantitatively, but dry red, it's a binary effect. You have these very few cells in the organoid that are blasting red, and these are actually the stem cells. So you don't have, this is a metabolic gene, this is not a developmental marker. I was expecting that we will see changes in levels with stem cells expressing some more, but what we actually see, this is an on and off effect. You have very few cells that are blasting in glycolysis, everything else is silence. So we decided to characterize this a little more. The intestinal stem cells, the LGR5 positive cells, are rapidly proliferating cells. We ask why these unique cells are doing work. And we knew from cancer biology that cells that are rapidly proliferating do work to increase their biomass. So they can have some extra metabolite from glucose for biomass production. So LGR5 positive cells that Hans Kleber described are actually rapidly proliferating. That's one different from other somatic stem cells. So we said maybe these LGR5 positive cells, because they are rapidly proliferating, they require glycolysis to maintain this proliferation. So we measured to see, are these unique highly glycolytic cells rapidly proliferating? And that's actually, we got another surprise. We stained for a marker of proliferation called Ki67, and we co-stained with this PDK1 cherry reporter. And we saw basically mutually exclusive staining. Cells that were green were not red, and cells that were red were not green. We didn't get a single yellow cell. That for us was a surprise. We said, hold on, we have this highly glycolytic cells, but they are not proliferating. And we know the LGR5 positive cells are rapidly proliferating. So what are these cells that are glycolytic and not proliferating? So we had to go back, read the literature, and we realized that actually in the literature there were several reports of a precursors of intestinal stem cells called the +4 stem cells. These +4 cells that sit here in the crypt, they are quiescent, they don't proliferate, and they are critical to regenerate the stem cells when you have damage in the crypt. So we actually stain to see, and we believe that the cells that were these highly glycolytic are these quiescent cells sitting here and important to regenerate the crypt when you have damage. So we needed for this to develop an antibody to work in the normal intestine. All I showed you was in the organoids, but we couldn't do this reporter system in the in vivo, in the mice. So we developed a good antibody that recognizes the substrate of PDK, phospho-PDH, and you can see, indeed, in the crypts, we have this single cell per crypt. You can see it here, 1, 2, 3, 4. Per crypt, we have one cell, highly glycolytic, same with the immunofluorescence, and we believe this could be the +4 cell. We confirm that this is not the LGR5. You can see here, we stain with LGR5 and phospho-PDH, and again, all the green cells that are the LGR5 are not staining with PDK or phospho-PDH, and you see the position 4 blasting here on red. Again, suggesting that this is a quiescent cell and may be important to functionally regenerate the intestine when you have damage. To prove that, we turn to an experiment where you can irradiate the animals. Okay? So you can irradiate the animals, the crypts will be wiped out, and the cells start regenerating. So you can measure levels of regeneration by measuring Ki67, a marker of proliferation. And you can see in control cells, you get a very nice proliferation. But if you treat the animals with dichloroacetate, so you inhibit glycolysis, you significantly inhibit the ability of these cells to regenerate, suggesting that indeed these highly glycolytic cells are there to regenerate, and if you inhibit glycolysis, the cells cannot regenerate. And we believed that when you remove SIRT6, you increase the number of cells, you have a better ability to regenerate stem cells, and unfortunately, when one of those stem cells acquired a mutation like APC, then you are favoring a tumorigenic phenotype because you have cells that are already grown to be glycolytic. So that's basically where we are on this project. We believe we identified a unique population of quiescent intestinal stem cells that are critically highly glycolytic. We still don't know why they are glycolytic. These are quiescent cells. Why they benefit from high glycolysis, we still don't know. But having this increasing glycolysis, if you have an increasing glycolysis, like in the absence of SIRT6, you may push self-renewal, and having increased number of these stem cells, by the time you acquire an APC mutation, you are basically promoting tumors because you start with a population that is better fit metabolically. And that's where we are basically with this project. The second question we explore, and I don't know how much time I have, but in a few minutes I will tell you.
Yeah, go ahead.
We are interested, is this really a phenomenon that only occurs in colon cancer? Is SIRT6 a tumor suppressor only in colon cancer, or maybe other tumors also care about SIRT6? So the one cancer we looked was pancreatic cancer. For multiple reasons, we have data from human samples where we saw that the same as in colon, in pancreatic tumors, we saw SIRT6 downregulated significantly in many tumors. So we asked, maybe in pancreatic cancer, which is a tumor that's up today, it's one of the most lethal tumors that we have. There is basically no good treatment for pancreatic cancer. They are extremely aggressive. Patients that are diagnosed, 95% of these patients will be dead in the next 1 to 2 years. It had a very low survival rate. We still don't have any new targeted therapies as we had for other tumors. So we thought maybe if we can understand better the biology, we could contribute to try to understand potential better therapeutics. So we decided to ask, using mouse models again, whether SIRT6 could be acting as a tumor suppressor in pancreatic cancer. And so we crossed animals that were mouse model of pancreatic cancer with either KRAS mutations in the pancreas alone or KRAS P53, which are both tumor models. The KRAS alone almost never give you full pancreatic adenocarcinoma. The KRAS P53 develop tumors with a latency of 25 weeks. In both cases, when we remove SIRT6, the animals develop extremely aggressive pancreatic cancer. Very rapidly. You can see even in this model where they already developed tumors, we aggressively, the animals came down with disease, some of them starting as early as 10 weeks. And all of these tumors, many of them have this lung and liver metastasis, which you rarely see in the normal model of pancreatic cancer. So again, as we saw in colon cancer, SIRT6 here was acting as a very robust tumor suppressor. And when you remove SIRT6, those tumors become much more aggressive. So the next question was, is this dependent on glycolysis? The same as in colon, maybe removing SIRT6 made those tumors more Warburg, more glycolytic, and that's why they were more aggressive. Now, I have to step back and tell you something. 95% of pancreatic tumors are driven by a mutation in KRAS. It's the most frequently mutated oncogene in pancreatic cancer. And there were several papers showing that the mutations in KRAS make pancreatic tumors highly glycolytic. So we were hesitant. We said, if these tumors, because of KRAS, are already glycolytic, why they will care about SIRT6? Why they will need to remove SIRT6 if they are already very glycolytic? But we have the human data suggesting that indeed these tumors downregulate SIRT6. And we got these mouse models where we clearly see that SIRT6 is acting as a tumor suppressor. So we decided to explore, we checked to see. Long story short, we tried to see if these tumors were more sensitive to glycolytic inhibitors, the same as we saw in the colon cancer. We treated these animals with the DCA drug, and we treated cancer lines that we developed from the animals with genetically knockdown of some of the glycolytic genes. And we saw no effect. So having or not having SIRT6 did not change the sensitivity to glycolysis. These tumors were already glycolytic, but taking out SIRT6 didn't make them more sensitive to glycolytic inhibition. So what was going on? Why these cells care about taking out SIRT6? And the answer came from stepping back and looking at the biology. SIRT6, as I mentioned originally, it's a chromatin factor. It sits on many genes. It represses many genes. It not only controls glycolysis. So we use a system where we did genome-wide chromatin immunoprecipitation to the mark that SIRT6 recognized, called H3K56. I mentioned SIRT6, the acetylate K56. So we took tumors from the wild type and the knockout animals. We did ChIP-seq for this mark, and we looked for genes that were specifically with increased acetylation in the knockout. And we came out with a list of 180 genes. And here is, you can see very nice, compared to wild type, the increase in the mark, the K56 mark, in one of the genes. And the top gene there was called LIN28B. For us, was a surprise. This is actually the mRNA levels, comparing wild-type tumors with knockout tumors, and you can see a 400 to 600-fold increase in levels of LIN28B. This was a massive deregulation of this gene, so we decided to explore further. What LIN28 is a gene that is expressed very early during development. It's what we call an oncofetal protein. It's expressed only during development. You can actually substitute MYC, the Yamanaka factor, when you are doing induced pluripotent stem cells. You can put LIN28B and you will get induced pluripotent. So it's really a very strong driver of stemness. Maintains stem cell signatures, fully silenced in somatic tissues. So we investigated maybe SIRT6 is an important chromatin factor to repress LIN28. And in the context of the pancreas cells, if you are a pancreatic tumor, by taking out SIRT6, you desilence, you repress LIN28, and maybe this is important to drive an aggressive phenotype. It has never been studied, and there were few studies in other cancers, liver and colon, showing that reactivation of LIN28 could drive cancer. In pancreatic tumor has never been described before. So we look, and just to make, I need to move this a second because I want to show you. Basically what we see is that if you are a pancreatic cancer line that have high levels of SIRT6, meaning LIN28 doesn't play a role, if you inactivate LIN28, you don't care. The cells keep growing. But if you are a human pancreatic cell line where SIRT6 is very low, this genetically drives an increase in LIN28, and if you knock down LIN28, the cells are dead in 24 hours. So you compare here the control with the knockdown LIN28, and you can see that the control keep growing, the knockout are fully dead in 24 hours. So those human lines that downregulate SIRT6 become addicted to the increased level in LIN28B, and if you inhibit LIN28, the tumors cannot grow. So lastly, the last part we said, okay, is this important in human patients? So we did immunohistochemistry in human patients, and what we found by staining for both LIN28 and SIRT6, that 30% of pancreatic human adenocarcinoma stain with this pattern of low SIRT6, very high LIN28. So we identified a novel pathway that is epigenetically driven, okay, by SIRT6, where if we can know that these pancreatic tumors have low levels of SIRT6, you can predict that these will be tumors that may respond well to inhibition of LIN28. And we are currently collaborating with several groups in other universities to try to develop LIN28B inhibitors, because we believe it could provide a major benefit to a high fraction of patients with pancreatic cancer. And that's basically where we are with this. We published this story last year, where we shown that indeed SIRT6 act as a critical repressor of pancreatic cancer by modulating level of this protein called LIN28B. Now, this gave us a very nice example of what we call epigenetic plasticity. Okay, I believe SIRT6 provide a beautiful example of what is called epigenetic plasticity, because in colon cancer, getting rid of SIRT6 gave these colon tumors an advantage, metabolic advantage. They were pushing Warburg effect, and this gave those tumors an advantage. Pancreatic cancer, they didn't care about that. They were already very glycolytic, but by removing SIRT6 as a chromatin factor, they got something else. They got repression of another gene, LIN28, and that provides those tumors with a unique advantage. So that tells us why tumors in general benefit from modulating chromatin factor. They acquire a plasticity to go whatever that tumor will benefit from. And that's probably why we keep seeing more and more this high frequency mutation of chromatin factors in cancer. And I think SIRT6 provides a beautiful example for that. So that's basically a summary of what I told you today. This is what we've been doing. We've been learning a lot of biology by studying this unique chromatin factor SIRT6. And I think we can still be learning a lot. It was a very good starting point. By looking at SIRT6, we are learning about chromatin biology, cancer metabolism, and the link between metabolism and epigenetics. So that's basically where we are, and I think I'll just do this. I will just show you. These are basically all the people in my lab that did this work, a lot of very talented postdoc and students, and many collaborators from different places. This is just a picture of the people in my lab here. This is basically the lab. That's the different people I was mentioning throughout the presentation. And with this, I will stop if you have some questions.
Sure. Thank you so much. If you can unshare the presentation.
Yes.
Great. It is a very good presentation. I really enjoyed it.
Thank you very much.
I have a couple of questions. So there are other students. Do they do the similar thing, or like they have
Actually,
different?
no. The lab now is it's diverse a lot in terms of projects. I have now some people working on a relationship of chromatin and DNA repair. I train as a chromatin biologist and with questions in DNA damage and DNA repair. And I have another postdoc working specifically on novel chromatin factors that we identify that are important for repairing DNA double strand breaks. This is one side of the lab. Another student is working on squamous cell carcinoma. We found that in skin cancer, this glycolytic switch is also very important. And we found that SIRT6 could act also as a tumor suppressor in the context of skin cancer. I have another student working on a completely different story that has nothing to do with SIRT6, where we look at metabolic liabilities. We took tumor cells, we put them under very stringent nutrient conditions, and we analyze how the tumors manage to survive and proliferate under these extreme nutrient conditions. And we have a very interesting story where we see that in the absence of some major metabolites like glutamine, which tumors need glutamine to survive, we manage to get tumors to grow completely independent of glutamine. And this is because they rewire metabolism in a very unique way that we are following now. So we're still asking a different type of questions, some related to cancer, some not. I have another postdoc working on very basic mechanism of transcription. We found that SIRT6 as a chromatin, the acetylase, does not regulate gene transcription by sitting on the promoter, but rather working intragenically with the polymerase. And we have a full story where we identify SIRT6 on a unique pathway that's controlling what is called transcription elongation. So that's a very basic project in the lab. That's nothing to do with cancer, or it's really a transcription. So the lab is very broad. We have some people working on basic questions, some people working on cancer.
And SIRT6 is been like mutated or anything in the cancer? Like, do you see anything?
It is. Actually, we found in tumors, it's most of the tumors downregulate SIRT6 at the level of expression. But when we look at the TCGA dataset, a few years ago, we identified 8 mutations in SIRT6, and we went on to prove that each one of those mutations were selected for. All of them either decrease the activity or the stability of SIRT6. We published a small paper in Cell Reports that you can look at to see those. And actually, this was already out of date a few years ago. I checked recently, and there are now more than 70 mutations described in SIRT6. So the list is growing, and the more cancers are sequenced, I believe we will see more and more of these mutations in SIRT6.
So I think that's about my questions. So I really appreciate. Thank you very much. It's a very wonderful presentation.
It's been a pleasure. It's been a pleasure, Sashiva, and I'm happy to participate and tell you about our work.
Yeah. So if you have some new stories in the coming months and all, if you want to talk about it, please feel free to.
I will. Thank you very much.
Give me a shout out.
Thank you.
Thank you.
Bye-bye.
Bye.